Hello, everybody. I hope you're enjoying your brief break as I'm off at a meeting. But what I thought I'd do is talk a little bit about the process of red blood cell formation. Now, we're going to talk a little bit about this more on Monday, but I just want to go over some basics. So in part one, we're going to talk about some basics that are involved in stem cells. So some things we talk about when we talk about stem cells. And then in phase two, I'm going to undergo specifically the lineage of cells that are in the blood. Right? So this whole process we're talking about, producing cells that are found in the blood, whether they're red blood cells or white blood cells, is called hematopoiesis. Right? It's a little tricky because we've got this extra E here for some reason, but this translates literally as the process of blood cell formation. So we're talking about hematopoiesis. Now, when we're talking about hematopoiesis, we're talking about stem cells. You may remember from general biology that we have cells that undergo cell phases where they're going to be born, start replicating their DNA, and then they become quiet for a while, and then they split in the process of mitosis. Now, that occurs in some cells, but most cells in the body do not undergo mitosis. They're born, and then they basically arrest early on in the cell cycle. They don't necessarily even have to copy their DNA. But there are certain cells that do this all the time. Now, sometimes these cells are doing something bad, like when we have cancerous cells, they're running out through the cell phases too quickly when they're really not supposed to be. But we have many areas in the body where we are going to undergo mitosis many times, and that is going to provide a steady stream of cells that are going to renew the body. So if we think about areas where the body constantly loses cells, like the skin, or the lining of the stomach, or the lining of the small intestine, there we have stem cells constantly producing new cells. Other places where there are fewer instances of injury, so for example, somewhere in, let's say, a tendon, right? You don't really have as many active stem cells because those areas tend not to be as injured as often. So we're talking about stem cells. These are cells that give rise to other cells. And when we're talking about stem cells, there are a few terms that we want to keep track of. One of these is potency. And essentially, this is a measure of how many different cell types a particular stem cell can give rise to. And so you usually hear when we're talking about stem cells in the news in the context of, for example, of embryonic stem cells, mostly what they're talking about is things that are pluripotent. Pluri means all, right? So these are cells that have all potency. So they can make basically every cell in your body. So that cell can divide, and under the right circumstances, the daughter cells over a period of generations could become basically anything. Could become nervous tissue, muscle tissue, connective tissue, right? Basically any cell you can think of in the body. Now you might think that's the most powerful stem cell that you can imagine, but it's not. Because above that, we have something that's more potent called a totipotent stem cell. And toti, you get the word total. And a totipotent stem cell not only gives rise to all the cells that are in your body, it also gives rise to cells that are genetically you but aren't currently in your body. So if you think back a long time ago to your birth, when you came out, right after you came out a placenta. That placenta was genetically identical to you, right? We could get your genotype based on that placenta, and is a tissue that originated for your embryo. So a totipotent stem cell can give rise to not only things in your body, but can also give rise to things like the placenta. You lose that ability to make a placenta very early, and so you very quickly stem cells quickly become pluripotent. Now, below pluripotent, we have stem cells that are multipotent. And a multipotent stem cell gives rise to the several different cell types that are found within a tissue, usually within a local area. So, for example, the stem cells we talked about in the small intestine were multipotent. They could give rise to some goblet cells. They could give rise to the variety of endocrine cells we talked about. They could give rise to the enterocytes, those cells that have the brush border and are going to do absorption. they can't become muscle cells, right? They can't become the smooth muscle of the small intestine. They can't become a skin cell. They're limited to 4 to 5 different types of cells that they can become. And again, they're in a particular area. When we have a cell that is multipotent, it's usually living in an area and it's going to produce stem cells for that area. That area is often called a niche or a niche. Okay. So below multipotent, we have unipotent stem cells. And a unipotent stem cell only gives rise to one particular type of cell. So it might give rise over and over and over to a squamous cell. So if we look in the skin, for example, the cells in the stratum basal, the stem cells that we talked about, are stem cells that only give rise to squamous cells, right? And those squamous cells are, of course, going to mature and move away from their niche, right? So that is a unipotent stem cell. It's not going to give rise to the melanocytes. There's a separate stem cell for that. It's not going to give rise to the erector pillae, right? That's a separate thing. So that's a unipotent stem cell. So the stem cells we're talking about in hematopoiesis are going to be stem cells that are multipotent, right? They're all involved things that are going to be found in one tissue. But that process is going to be fairly complicated. So this is a picture, actually it's a simplified picture, of the cell tree, or what is called a cell lineage, of hematopoiesis. So a cell lineage is essentially like a family tree for a cell. So we have a cell at the top that is going to be the first cell of this stem cell. It can become many different types of stem cells. It gives rise to daughter cells, right, which give rise to other daughter cells, and so forth. And you'll notice on this tree that we come to some points at which we have a cell that is a stem cell that gives rise to several different types of cell, and then those cells are the mature version of the cell. So, for example, an erythrocyte right here is a mature red blood cell. Right? So that basically is the end of the line for that particular part of the lineage. If we go over here to the myeloblast, right, that is still a stem cell, because you notice that there are still different points at which that cell can mature into other cell types. Right? So it's still a stem cell. It can give rise to multiple types of cell. Now, when we're talking about this graph, there's something else that's interesting, and that is that at some point, a stem cell loses its ability to give birth to anything other than one particular type of cell. Right? Now, there's usually not shown on this graph very much, but let's give a look to, for example, this common lymphoid progenitor cell. This is a stem cell that can give rise to a natural killer cell and a small lymphocyte, so 2 different types of cells. So it is still a stem cell. It has the possibility of making multiple cell types. If we look at something like this common myeloid progenitor, still very similar, right? Still gives rise to multiple cell types. Between this step here, from going from that cell to the erythrocyte, there are still other stem cells that are not shown here. So we go from this common myeloid progenitor, which can give rise to several different cell types. One of those is going to be a unipotent stem cell, which will divide and divide and divide to produce only erythrocytes. Right? So it's a unipotent stem cell that only gives rise to these red blood cells. That's still a stem cell. It's unipotent, but it can only make one thing. So when we have a cell that started off with the ability to make many different things, but eventually gets to the point at which it can only make one cell type, that cell is said to be, I'll get a fresh piece of paper of this, committed. Right? It's no longer running around making all different types of cell types. It is committed to making one cell type. So it's a stem cell that has settled down to make a single type cell. So what we're going to do tomorrow, or I should say in the next video, is we're going to take this and we're going to apply this to the process of the formation of blood cells. And we're going to see how some of these ideas, the ideas of the cell lineage, the ideas of being multipotent versus unipotent, and the idea of being committed are going to play out in that particular lineage. Welcome back to part 2 of our video on hematopoiesis. Again, that's the process of blood cell formation. So we're talking about the rise of the blood cells, and we're going to try to use some of those terms we used in the last presentation in the specific context of blood cell formation. So as you may recall from last semester, red blood cell formation, in fact all blood cell formation, occurs in the red marrow. And the red marrow, of course, is that spongy reddish material, that occurs in the heads of long bones and a few other places in the body. For example, the body of your vertebrae, the centrum of the vertebrae, contains an area of blood cell formation. The hip bone contains some areas of blood cell formation. So, of course, the hip bone is a flat bone, but it does contain some areas where we can make red blood cells and other cell types. So, our red marrow, again, we're going to look at long bones because that's the most common place where we'd form blood. And within that, we have this red marrow. And if I look more closely at the red marrow, I would see that it was filled with something called sinusoidal capillaries. And the word sinusoidal means sinus-like. And so if any of you have ever suffered from sinus headaches, right, what we're talking about is headaches that originate from inflammation in your sinuses. And so these are hollow places within your skull, and that should be the key, hollowness, right? So a sinusoidal capillary would be running through this system. A capillary, again, is the smallest vessel we have. Usually they're a simple tube, but in a few places, including the red marrow, they're sinusoidal, which means they have large areas that are hollow, right, where the cell tube breaks down, and we have an area that is essentially, we could look right into this tube. And surrounding the sinusoidal capillary, we have stem cells. So I'll draw a few stem cells here. And they're constantly dividing. And so here we have one giving rise to 2 daughter cells. Here's another one giving rise to a daughter cell. It makes a copy of itself. Okay, so we have all this stem cell activity occurring outside the sinusoidal capillaries. And now, because we have these sinusoidal capillaries, some of those cells, once they reach a particular maturation state, can travel through our little window and enter the capillary. And from that point, they're in the bloodstream. So they will move out of the red marrow, travel around the body until they die. So we have an area where we can have a lot of blood cell formation. We also have this process for them to leave the red marrow and travel around the body. So let's take a look at some of the cell types that would be produced in the process of hematopoiesis. So our cell types, we all know some of them already. We have red blood cells, also called erythrocytes. So these are cells that are going to carry oxygen around the body. We have a whole variety of white blood cells. And white blood cells are involved in a variety of immune responses. And we'll see in class there's several different types of white blood cells. We're going to talk, there's a big class called lymphocytes, which we'll talk about mostly when we talk about the immune system. And then there's a whole other class that are simpler that we'll talk about within the next week. We have the megakaryocyte. And as this name suggests, this is a large cell. And this cell will give rise to platelets. Right? So it's a large cell that breaks down and forms platelets, which are in fact little dead pieces of cell that break off the megakaryocyte and then float off into the blood supply. We also have within this group sort of a mysterious set of cells that are going to help make the osteoblasts. osteoclasts. Right? And so this is a little bit mysterious. We do know that the red marrow, there are some cells that are going to be part of making an osteoclast. They fuse with other cells from various other areas. This process is not all that well understood, but suffice it to say, if you don't have any red marrow, you're not going to make any osteoclasts because that is necessary for osteoclast formation. So those are major points. We're really going to be focusing in on the red blood cells today and a little bit on the white blood cells. And what I expect you to know mostly for the test is what's going on with the red blood cells, because we'll talk about the whole process of their maturation in quite a bit of detail on Monday. So we're going to start off and talk about the cell lineage of the red blood cell and a few other cell types. And then a little bit later, again in lecture, we're going to talk about how actually, once we kind of get to the point at which we have cells that are going to become red blood cells, how they actually go about maturing. So we're going to start off with the cell that is at the top of the lineage. And so you can think of this as sort of the great-great-grandfather of all blood cells. So this is known as, and we're going to start off way back in time, a long-term, we'll call that LT, hematopoietic stem cell. Now, that's a lot to write down. So this is usually just called an HSC for ease of writing. So this is a long-term HSC. I should also say this is also sometimes called the hemocytoblast. So yet another name for it. So on the test, I would expect you to be able to call this a hemocytoblast or an HSC. Now, the reason this is called long-term is this is a cell that exists very early in development, and it is pluripotent. So stop and think about, what does that mean? Well, it means that it can form any cell in the body, anything that's not the placenta, for example. So this is obviously a very potent cell. Fairly quickly, this is going to mature into what is called a short-term HSC, or a short-term hematopoietic stem cell, and this is a multipotent stem cell, which means it can no longer just become anything in the body. It instead is going to be basically just able to form blood cells of varying types. So this is what we're really going to focus on. These guys are fairly interesting, right? They are multipotent stem cells. They don't just live in the red marrow. It turns out they can jump out of the red marrow, travel around the body for a bit, and then go enter another red marrow. So if you were to follow one of these through a slice span, you would see that it would leave, for example, the red marrow that's associated with your femur, leave that, hang out for a bit in the bloodstream, and then go take up residence again in a red marrow that's associated with your vertebrae. So there's ways to collect these from your blood just by looking for these transient short-term hematopoietic stem cells that are going from one area to another. So that is our short-term hematopoietic stem cell. We're going to focus in on that cell and its progeny, or its children. So we'll start at the top again to give us a little bit of room. And we have our short-term hematopoietic stem cell, which again was multipotent. Now, this cell can divide and give rise to other types of stem cells. And so we're going to talk about two different cell types it can divide into. So it can produce something called the myeloid stem cell, or something called the lymphoid stem cell. Both of these are also multipotent. So I'm going to put another M next to them, indicating that they could become several different cell types. Now, we're going to talk about the lymphoid first, because this is going to be fairly simple. So the lymphoid turns into lymphocytes. And again, these are cells that have immune function. We will talk about them when we talk about the immune system. These are things like T cells and B cells. They also can divide and develop into dendritic cells. Now, not dendritic as in a dendrite that's in the central nervous system. These are those cells that are also called the Langerhans cells that live deep at the layer of your skin. So they eventually migrate away from the red marrow and take up residence in the lower epidermis. So again, we have the cell type. It can give rise to two different cell types, so that's why we call it multipotent. Now, we're going to focus a little more on the myeloid side of things. This stem cell can give rise to basically two major pathways. One of these is going to give rise to the myeloblast. This is also multipotent. And so this cell gives rise to a wide variety of different types of white blood cells. Essentially every type of white blood cell other than the lymphocytes. So we're going to talk about basophils, eosinophils, and things like that in lecture. They all come from this myeloblast. So this is multipotent because it's giving rise to multiple stem cell types. Or I should say giving rise to multiple cell types. The myeloid stem cell can also give rise to something called the proerythroblast. Now, this cell is unipotent. And in fact, we're going to talk about this cell quite a bit in the lecture on Monday. So this cell is unipotent, right, which means it will still continue to divide, but every single cell type that comes from it is going to be of one type. So one of these, in a process of maturation, will eventually become a red blood cell, right? And we produce a lot of red blood cells. So every day you're making between 100 and 200 billion red blood cells. So an incredible amount of red blood cells. So you can imagine this myeloid stem cell is constantly dividing, constantly producing a lot of these proerythroblasts, and they are eventually going to develop into red blood cells. So hence unipotent. They're still stem cells because they still divide, but every time they divide, the only thing that's ever going to come out of that is a red blood cell. So I think we've addressed a little bit of our idea of multipotent ability to generate a lot of different cell types versus unipotent ability to generate a single cell type. There's one other thing I want to talk about, and that is the idea of commitment. Because it's beyond the scale of what we're doing here to talk about how this process occurs, but there are cell signals that allow each one of these cells to move from one stage to the next and choose to become a particular cell type when it divides. And understanding that is incredibly complicated, right? But there's a whole field of research that just tries to figure out what's the cell lineage and then how is that cell lineage controlled. Some of those signals, right, are going to allow a cell to become committed. And again, a committed cell has reached the point at which it will only form one cell type. So if we look at a line of commitment, somewhere where a cell has made a turn where it can no longer go back and become anything else or make more than one cell type, the proerythroblast, somewhere between the myeloid stem cell and forming the proerythroblast, has been committed. So it's gone through commitment. And so that is an irreversible step, and so there's a lot of interest in how those things occur. The myeloid, right, is not a committed cell. It could still give rise to either one of these cells. There are extreme cases in which you might be able to take the myeloid and with the right signals force it to become a short-term HSC cell again. And so there's a lot of stem cell research that's talking about that. You could never theoretically take a proerythroblast and convert it back to this myeloid or back to the short-term hematopoietic stem cell. So that's it for this particular video. Again, we will focus a lot on this process of maturation of red blood cells on Monday. So have a great weekend, and I will see you guys on Monday.